session so that you can refer to it later. So any reaction requires activation energy. The activation energy is needed for the reactants to turn into the products. Good. So, in order for this product to be formed, a reaction needs an activation energy. This energy is not related to whether the reaction is exothermic or endothermic. No. This activation energy is needed for the reaction to start. It's called activation energy. Okay? Now, let's take this example. This activation energy here is needed for this reaction in order to turn into product C. If we add an enzyme plus enzyme, if we add an enzyme to this reaction, which is a biological catalyst, the activation energy needed to make this reaction will be less. So instead, for this reaction to wait until it reached this point, the reaction can go this way, for example. So you see, this is the difference in the energy thanks to the enzyme activity. So we say that the enzyme lowers the activation energy. As a result, the reaction takes place faster. The reaction happens faster. So, let's say this amount of energy, look at this, the blue one. This amount of energy needed will take, let's say, 10 hours, but this amount of energy can be reached in 2 hours. So when we add an enzyme, the reaction will take place in 2 hours because small amount of energy is needed. And one of the examples I gave before in term 1 when I explained about this is that old cars, during cold days, if you start the car at the morning, you need to wait, let's say, 15 minutes until the needle or the sign of the temperature rises to a certain temperature where you can start the engine and use the car. I used to have an old model car. At the morning when I want to go to the college, I have to turn it on. I have to wait 15 minutes until a sign goes up. Before the sign goes up, I cannot drive the car. Yes, I'm repeating it. So, there is a substance I can add it to the radiator. Instead of waiting 15 minutes, I can wait 5 minutes, then I can drive the car. This substance, it's like comparison, the same concept as the enzyme. It will lower the activation energy. Instead of waiting 15 minutes until the energy is needed, I can start the car after 5 minutes. No need to wait until that much to get that much heat. I can start the car with lower temperature. So this is how, chemistry-wise, how enzymes work. So now if you see all of you lowering activation energy, can you understand that? Now, the next structural characteristic of an enzyme make the enzyme very reaction specific. When I say structural characteristic, what do I mean by that? No, we are speaking about the enzyme still. What do I mean by structure? Structure, yes, shape, 3D shape. Yes, and what you said is right. Because an enzyme is a protein, right? And the function of the protein is related to their... Very good. The function of the protein is related to their structure or shape. And their shape is related to their 3D shape, which is related to gene expression, etc.
It's like a tertiary structure.
Yes, tertiary structure, very good. Now, yes, it can be quaternary. It can be quaternary.
What is it? Rare for it to be quaternary?
Yeah, most of them are tertiary, because usually quaternary structures are complex proteins. So, when we say specific, it means there is one enzyme for one substance. You cannot use the same enzyme for different substances. This is like the ID, or simply we describe it as the key lock model. It means this enzyme
Mister, can I ask you a random question?
yes.
Mister, which subject do you like to teach? Earth and space or biology?
Earth and space طبعا.
I like to teach.
Bio.
Actually, I'm fine teaching anything,
No,
but
like,
I
which one do you find more interesting?
Bio.
now,
Which class does he enjoy more?
to be honest, I've been teaching biology for 14 years. So for me, teaching biology is much more easier than teaching Earth and space. But I'm enjoying teaching Earth and space because I'm learning new things myself.
Earth and space أول سنة
Y- y- Yes.
طول عمره بايو. أصلا إيش حطوا Earth and space؟
Okay. So look here.
This the substrate is the solid and the active site is the compatible. If not, it won't be.
Can you repeat, please?
Yeah.
Not you, Fatima.
Physical والchemical properties need to be compatible.
Yes, exactly. Yes, Aisha, you can repeat now. Yeah. So let's finish this quickly, please, because it's obvious that this enzyme can only act on this substance, but it cannot act on this substance. So it's like key and lock. You understand that? They have to fit together. They have to match together. And when I say they have to match together at 2 points of view, the first one is chemical, the second one is physical. So when I say chemical, it means I'm speaking about polarity, I'm speaking about charges, same charges, polar, non-polar. And the other thing is physically. Physically, it means I'm speaking about fitting, the shape, or just physical.
Mister, like the video yesterday, you said physical and chemical properties of the substance.
Exactly. When I say chemical property, it means I'm speaking about the pH. They have to work in the same pH. They need to have the similar charge, similar polarity. Physical, it means their shape must match. They have to be compatible. We say compatible. So you see here, there is matching between this enzyme, this is the enzyme, and this is the substance or the substrate, we call it. They have to match. If they don't match, they cannot. And this is, and this is what we call it here, specific. Each enzyme is specific for one substance. The shape and charge of the substrate, I mean by the substrate, the substance that the enzyme acts on, must be compatible with the active site. This is the active site of the enzyme, the part that links to the enzyme. Okay? Enzymes are not consumed by the reaction, they are reused. What does this mean?
Because they can be used, they don't like
Yes, exactly. So, if I want to,
يعني مثلا kind of like lysosome because lysosome can like reuse something like that.
what?
Because they're not chemically changed during the reaction.
Yes. I don't add enzyme in the reactant or in the product. There is no place for the enzyme. So as if the enzyme is added somewhere.
It's like when you add part of the
Yes.
reaction.
It's not exactly. It's not consumed. It's not produced. But it will change its shape during the reaction. Then it will return back to its normal. So, let's say we start 1 minute with this enzyme having this shape.
And it happens somewhere in the middle. When
During the reaction, it might change its shape. During the reaction, it might change its shape. After the reaction, simply it will One minute, yeah. Yeah, I will explain it. So, we say that the enzyme the enzyme is not consumed during the reaction. So
That's why we can reuse it.
yes, exactly. So before and after, it's the same. I'm telling you, wait 1 minute. After and before, it's the same. But it can change during the reaction. I'll give you an example. Yes. Imagine you have a door that is locked. A locked door. Okay? Pay attention here. Okay. So, look at that. This is not the key. So, this is a key, right? As I pay attention, everyone, please.
Oh, high substrate وهاي مال
Yes. So, the key here, pay attention, please. So, imagine this enzyme wants to break down two substrates that are together. So, this enzyme is allowing hydrolysis to take place, or breaking down to take place. This is one substance, this is another substance. I cannot break down these two substances without an enzyme. This is the enzyme. So let's say the enzyme, it has to match and break them. After that, is there any change in the enzyme? No. And it will allow the reaction to take place. Enzyme is not an end product. It's not a reactant. It will only help separating these two substrates or joining them together. At the end, it's not used. But during the reaction, the enzyme is used. So it might change its shape temporarily. So when I put here, for example, the key now cannot be used because it's already in use. You get the point?
Yes.
During the reaction, the enzyme is being used.
Yes.
So you cannot use it in 2 or 3 different reactions. At the end of the reaction, the enzyme is free again.
Mister, what about
So the enzyme before and after the reaction is the same. During the reaction, it will change its shape in order to bring the two or separate the two substrates or to allow the substrates to combine together.
Mister, what about the cell?
What the cell?
Like, can we reuse it again or like no?
What do you mean by the cell?
Like cell.
Cells divide. So they reproduce. Cell, once it is damaged, it has a lifespan. Cells, okay. So let's say different. Sorry, cells divide. Some cells they stay for a long period of time, like neuron cells. Some cells they are renewed every few months, like blood cells. Yeah. Okay. Any question before I continue because we need to move? Yes, Aisha.
After a reaction, what do I write? It goes back to
It's the original shape.
It's just that
Okay, shall I continue? I need your attention, please. Enzymes are biological catalysts that facilitate chemical reaction in cells by lowering the activation requirements of activation energy. We said that activation energy is the initial energy. Girls, please. This is distraction. Activation energy is the initial energy required for a reaction to occur. We already explained this here.
Mister, we took enzymes and I think
Yeah, I explained those before. Now, let's answer those questions, and this is what we are going to study in the next one. So we finished 3.1. This was 3.1. Let's move to 3.2. Pay attention. What does it mean for an enzyme to be denatured? Look at this term.
I'm moving 3.2.
3.1 still?
لا ما بعرف 3.1 بعد. 3.3 not with me.
Yeah, 3.2 was the
3.2 was talking about in case they synthesize, digest, ما
Okay.
والactivation energy graph.
so
Type.
3.2 was.
I will double check because College Board is not opening.
I think we have something.
Okay, pay attention. Anyway, let's finish those. Then I will look into the College Board to see which lessons we covered. Denatured, it comes from two terms. Nature, denature. It means changing its nature. Exactly. Again, we go back to the first phrase I introduced enzymes with, which was treat enzymes as proteins. In order to know how enzymes are broken down or are inactivated, we have to think about how proteins are broken down or how proteins are inactivated. And we said that proteins are sensitive to changes in the environment. It means if you change the temperature, you might change the shape of the protein, you might change the R groups, how they bind. And if you change the pH as well, you can have changes in the protein.
So denature يعني it's like nature of the enzyme. We can
Yes,
change.
exactly. Sorry?
يعني denature it means like the nature of the enzyme can
Change,
change
yes.
to some
Yeah. So, pay attention. If I break, pay attention. If I break the top of this key, will it function the same way?
No.
It won't function. Whereas if I break the bottom of this key here, this side, will it work?
Yeah.
Yes. Because enzymes have active site. What is active site? It's the part of the enzyme where it binds to the substance. Not all of it will bind to it. So if I have an enzyme, so let's say this is an enzyme. This part here represents the active site of the enzyme. Or let me change the color. This is here the active site of the enzyme. The other one, it's only the body of the enzyme, but it's not needed for binding. As long as the active site maintains its shape, the enzyme can work. Okay? If there are changes in the active site, or what if, think about this, and this is coming next. Think about it. What if I block the active site? No, that's not denature. Imagine that I put something and I block the active site.
إذا حطيت inhibitor خلاص
ما بيشتغل.
It means, imagine I have this key and I wrap this key with glue or scotch tape. It's still there, but it won't work. It will be inactivated. You get the point? So the active site, it's where the enzyme will bind. If I block that, there won't be any binding. Okay? And this is coming in the next lesson. Now, no, this is inactivation. Denature, it means changing the whole shape or structure. How do... Yeah, like pH and temperature. We'll talk about that. So,
Can you write the meaning of it?
yes, don't worry. So, what does it mean for an enzyme to be denatured? Simply changing its structure, full stop. Changing its structure. When you change a structure, you will lose it because it is a protein and the function of the protein is related to its structure. If the structure changes, the function won't be able to be done. You understand that?
Yeah.
So, what does it mean for an enzyme to be denatured? It means that here, it means that the structure of the enzyme is changed.
What does denature mean? So
It means changing the structure, denature, changing its nature. Okay, like for example, this is the shape and it's like that. It's denatured. Okay? It's fine, you are paying for that. The structure of the enzyme is changed. Now, here we can raise another question. How we can change the structure of the enzyme.
I would say and temperature.
S-
ترى في raising hand. أكيد في raising.
Okay, Aisha, can you answer please?
Yeah, because we change the temperature and pH.
Very good.
Because
Treat it as protein.
And then how does the pH affect?
The pH will affect the binding of the R groups, the bonds that happen in the R groups in the protein. And it can break some bonds, changing the structure of the protein. Okay? So how? Simply by changing in temperature and changing in pH, acid, base, alkaline, etc. So, what happens at high temperature? So I'm drawing a graph here that represents enzyme activity as a function.
The code is in the lower pH.
Yeah, that's right, because the main idea of enzyme activity, you will see that physics is everywhere in life. The main point, yes, physics, because the binding of enzyme, mixing enzyme with substrate, all depends on something called thermal energy. The activity of the enzymes, and this thermal energy will change into something called kinetic energy. This kinetic energy allows molecules to move and they start hitting each other. So the lower the temperature, the lower the thermal energy, the lower the kinetic energy, the lower the chance they will bind together, the slower the reaction. So let's say in our body, what is the optimal temperature? 37.
38.2.
This is enzyme activity. Okay. Go. And this is here temperature. You are coming back or no?
No.
Okay.
Oh, I had 3 days.
I will share the notes later. Check them.
So in binding activity will be also lower, صح?
Yeah. So look at that piece and keep focusing. The temperature. The maximum temperature is going to be the optimal temperature of the body. Below that temperature, what will happen to enzyme activity? And above 37. So the relation isn't proportional. What do we call this type of graph?
This is obviously
Parabola?
parabola.
No, parabola. Hyperparabola is opposite. I'm not sure. Parabola.
This is like the opposite.
Okay. Anyway, so we have a maximum point here. This is the best point for the enzyme activity. And let's say here it's around 37 to 40. Okay? What will happen after that? As temperature decreases, enzyme activity decreases, right? And as temperature increases above 40, enzyme activity decreases again. Now, what will happen at this temperature? Let's say this one is 70 degrees, and this one here is 0 degrees.
You die.
What? It's not something living to say you die. Okay? No, who said that? No, we are speaking at 0 degree, the enzyme activity will be 0. Enzymes won't be working. Yeah, no, I'm not speaking about us, we are speaking about an enzyme. Yes, I know. I'm speaking about the enzyme activity per temperature. I'm saying they will work best at 37. For this reason, when you have a fever or drop in your body temperature, the metabolism in your body changes. Yes, yeah. The activity changes. Okay, pay attention. We will talk about that. So, girls, no. We'll talk about that later. Khulud, pay attention. So, guys, please. Side chats distract me. I told you this semester, sorry, but during this session I need full focus I'm not going to explain 50 minutes. It can be 48 or 49. Don't worry. Okay, so at freezing temperature, what will happen to the enzyme?
It will not gonna be able to bind that one, so there will be zero.
What will happen to it? It will be destroyed.
It will lower the binding activity of the enzyme.
will stop, it will hold its shape.
It will hold its shape. So at very low temperature, the enzyme does not break down. It's not destroyed. It is inactivated.
So it will not
They stay in the same shape.
it will have the same shape.
So it's not denatured. No, it's not denatured. Wrong. It means, hint, as if you are preserving food in the freezer for 10 days. It will still have the same shape. Okay? So, if I return that enzyme from freezing point to optimal temperature, will it work? Yes, it will work. Because freezing does not destroy the enzyme. It will only inactivate it by freezing it, preventing it from moving.
How about burning it?
Wait, calm down. For this reason, yes. For this reason, if you freeze raw meat without cooking it, if you freeze it, the raw meat, if you freeze food or raw meat for a period of time, then you get it out. There is a high chance that the bacteria that was there will divide again, because freezing won't destroy it, will preserve it. That's why there is a chance that a virus or a bacteria from thousands or millions of years ago can, when the polar ice melts, there might be new viruses coming, because those viruses can be preserved for millions of years. Okay? It means freezing temperature, preserve the shape of the protein. It means preserve the enzyme. Now, what happens at 70 and above? Don't say die for enzyme. It's not a living thing to say die.
It will not work.
Why? Denature or destroy it.
So the structure will change.
Yes, very good.
Okay, I have a question.
Yes.
Okay. The first session, the first class يعني, you said يكون الانزيم frozen لا when يكون normal temperature they are active.
Yes.
Okay.
Their activity is maximum. 100%. Optimum.
Their shape changes.
Sorry?
at
No,
normal
no.
temperature?
At normal temperature, the activity is maximum. This is the graph. This is the activity, the percentage. So here it's working 90%, the enzyme. As temperature increases more than 40, let's say it's here 50 or 60, the activity will be, let's say, 50% or 60%. Okay, at degree 20, it might be 40%. At 10, it's 10%. At 0, it's let's say 0%. The enzyme is not working at all. You got the point or no? Optimal temperature, it means the good temperature, the best one. So the best one in our body is 37 degree. At 37 degree, the enzyme, it's having its shape, it's not denatured, not inactivated, its efficiency is maximum. It means the enzyme is working very well. It means that you eat food, whatever happening in your body, the enzymes in your body are working right away. In digestion, and cellular respiration, and any other reaction in your body. If the temperature moves away from the optimum, either more or less, the enzyme activity will start dropping down. So if you have a fever, 41, 42 degrees, your enzymes won't work very well. So even you won't be able to digest well, you won't be able to have energy well, because your enzymes are not working at their maximum efficiency. Is that clear?
Yeah.
Yes.
Mr. Now, if the temperature was like at 70 and then it dropped to 57.
Yeah.
So
Very
then
good
what
question.
about,
Very good.
خلاص it's destroyed.
I will give you +1 on this question. +1 to Mariam. Because I want, yes, Mira will answer.
خلاص it is destroyed.
Okay, Mira took that plus.
It
No. She will
0.5.
Okay. The question is, now after the temperature is 70 degrees,
It will be denatured.
you return back the temperature to normal. Will the enzyme work? No. For this reason, you can cook food, then you can eat it after that. Yes, exactly.
The chemical composition of the change.
Yes, exactly. Okay.
If it's at 0 degrees, it
Yes.
can't go to the middle.
Yeah, because here there is no change, and
What's your last name? Is there another option?
Here, this class.
No, I checked it.
yeah.
Experimental treatment is different than experimental treatment is the treatment they're testing here. The treatment is just general treatment with government.
Speaking about something called control group in the experiment. You can control group in this. We don't expose it to the experiment. That's it.
Oh,
Okay.
okay.
It's not part of the experiment. Like, for example, I'm going to experiment on plants, and one of the plants captured this light. I cannot change it.
Can I write, like I write normal and I take a picture of this and
Yes.
post it?